In this video lecture, we will be covering chapters 27 and 28, which are about the reproductive system. So chapter 27, specifically, it's about the male reproductive system. So the reproductive system consists of primary and secondary sex organs. So the primary sex organs are known as gonads, and they help to produce gametes. In the case of the testes, it will produce sperm cells. In the case of the ovaries, oocytes. And they also produce hormones like testosterone in the case of the testes, and in the case of the ovaries, it will produce estradiol or estrogens and progesterone. Now, the secondary sex organs are the organs that are necessary for other organs that are necessary for reproduction. And in the male includes the ducts, the glands, accessory glands like the seminal vesicles, prostate gland, the bulbourethral glands, and their function is to produce these fluids that helps to deliver the sperm cells into the female reproductive tract. Now, the female secondary sex organs will be the uterine tubes, also known as fallopian tubes or oviducts, also the uterus and the vagina, which is the copulatory organ in the females that receives the sperm, and also helps to act as a birth canal. Now, also part of the reproductive tract, reproductive system structures will be the external genitalia, that it is located in a triangular area between the thighs that we call the perineum. Now, in the males, the external genitalia will have the testicles and also the penis. Now, internal genitalia will be the genitalia that is located within the pelvic cavity. So, except for the testes and some associated ducts in the scrotum. In both sexes, the secondary sex characteristics are features that helps to distinguish the sexes and influence mate attraction. These secondary sex characteristics develop at puberty under the influence of the different hormones like testosterone, estrogens, that in both sexes makes the pubic and the axillary hair to develop in these areas, and also these glands that are in the skin of these areas become more active, and then you will have change in the pitch of voice for the males. And then in the males, you will have facial hair developing. Also, you will have visible coarse hair in the torso, the limbs, and increasing the muscular mass. For the females, these secondary sex characteristics will be influencing the distribution of body fat. So we'll have fat around the hips, and also in the anterior area of our pelvic area, or pelvic part of the anterior wall of our body. And also it will influence the deposit of fat within the pectoral area, so we have enlargement of the breast, and in general the skin is going to be thinner and it's going to have less hair than the males. Now, genetic sex is determined by the presence of the sex chromosomes. So it will be XY in males and XX in females. So 2X chromosomes in females and only 1X in males. And the absence actually of the Y chromosome will be determining female genetic sex, because there might be some conditions in which the male only has one X chromosome instead of having two. And in that case, since the female doesn't have the Y, that will you will have absence of the Y chromosome. In that case, the person will be a female. Now, sex is determined by the type of sperm that fertilizes the mother eggs. So the egg of the mother is always carrying X chromosomes, but the male uh, has the X or the Y. So if the sperm cell has the Y chromosome and fertilizes the egg, the product will be a male. And then if the sperm cells carry the X chromosome, then the product will be a female. And you can see that in this diagram. Okay, so in here, how this sperm cell has the X chromosome, so it combines with the X chromosome from this egg, so you'll have a female. And then in the case of this Y chromosome in the sperm fertilizing the egg, will make a male. So the external genitalia, and in general all the genitalia, develops in utero in the proximity of week 6 of development, and all of these structures come from this genital tubercle, the uterogenital fold, and labioscrotal fold. Now by week 8 there is no differentiation between male and female, but it is after that, especially during week 12, where you have actually development of the male or the female genitalia. So in the case of the males, they will have developing of the glands of the penis out of this genital tubercle, and then you will start developing this scrotum and the testicles out of the labioscrotal fold. And then for the females, you will have analogous structures like the glands of the clitoris in females, labia minora out of this urogenital fold, and labia majora out of this labioscrotal fold. And then by week 12, you have pretty much well developed what is going to become the external genitalia in males and females. So the gonads, since they develop in the abdominal cavity, they will migrate into the pelvic cavity for the ovaries, and in the scrotum outside of the body for the testes. In order for this migration to happen, there will be specialized connective tissue that is called the gubernaculum, that it will bind inferiorly the testes in males and in females the ovaries. As time is passing by, this gubernaculum is going to shorten, and then it's going to bring the testes into the scrotum, and it will bring the ovaries into the pelvic cavity. Now along with this, you will have also a process that is called the vaginal process, that is a fold of peritoneum that it will send into the scrotum, that surrounds these testes, protecting them in their migration into the scrotum, passing by a pathway that it is called the inguinal canal. And this inguinal canal it is the most common site of hernia in males. So the testicles begin descent into the scrotum as early as week 6, and then by month 7, it's supposed to start passing through the inguinal canal and then descend into the scrotum. Now, as the testes are descending, the testicular arteries and veins, lymphatic vessels, nerves, and spermatic ducts will be also descending. If the male doesn't have the testicle descending, this condition is going to be called cryptorchidism. And this affects around 3% of the male birds. Usually it's unilateral, but it can be bilateral. And then if you have bilateral testes that I have not descended, you can perform surgery right away. But when there is a unilateral testis that doesn't descend, you will wait a little bit to see if it descends by itself. If not, you perform surgery. Why is that? Because spermatogenesis, or the production of sperm cells, has to happen at a lower body temperature, like 1 degree Celsius less than the normal body temperature, which is around 37 degrees Celsius. And actually in the abdominal cavity, the temperature there, the intra-abdominal temperature is greater than the actually 37 degrees Celsius. And when these testicles, their tissue that forms the sperm cells, are subject to this high temperature, the rate of mitosis of the stem cells that makes the sperm cells is so high that it can create mutations in these cells, and then the male has the chances of having testicular cancer, or you can have infertility. Now, before we move on, let me talk about the key points for both the female and male reproductive system. So, within the males, there is this gland that surrounds the urethra and that produces an alkaline fluid. This is the prostate gland. Now, within the males, there is some, in the testicles, there is these nerve cells, like nerves, the nerve cells that coordinate spermatogenesis. These cells secrete a hormone that is called inhibin, and they are found in the seminiferous tubules within the testes, and they form a blood-testis barrier. Now, the special type of cell division that is required to produce gametes is called meiosis. Now, within the testicles, there is other type of cells that are called interstitial cells that helps to produce testosterone, respond to luteinizing hormone, or LH, and these cells, interstitial cells, are also known as Leydig cells, and they are located between the seminiferous tubules. Now, for the female reproductive tract, there is this phase of the menstrual cycle that is called the secretory phase. And in this secretory phase, the corpus luteum is formed. Now, the uterine phase that develops because of the fall of progesterone levels in females will lead to the menses. And the main hormone that it is secreted by the corpus luteum in females is the progesterone. Now, some of the actions of estrogens is to stimulate bone growth, to maintain accessory reproductive organs, and also to maintain female secondary sex characteristics, and also initiates the repair of the endometrium. Now, for fertilization, the segment of the uterine tube where fertilization happens is the boundary between the ampulla and the isthmus. Now, both testes and ovaries respond to luteinizing hormone. Both produces gametes, and in both meiosis takes place. And also both respond to follicle stimulating hormone. The other key point is the function of the ovaries is to produce oocytes, to secrete a hormone also that is called inhibin, to secrete hormones, and also to form immature gametes. After ovulation, the ovary will secrete both estrogen and progesterone. Okay, so those are all the key points. So then this is pretty much a diagram to show you how the testis descends. So here in this green color you can see the gubernaculum, that it is attached to the inferior pole of the testis, and as the time passes by between month 3 in fetal life up to month 8 in fetal life, this gubernaculum shortens, and then as it's shortening, it's pulling the testis through the inguinal canal, and then it brings it into the scrotum. So in the infants, or in the babies once they're born, the testis is supposed to be descended into the scrotum. Now this is a mid-sagittal view of the male reproductive system. So the male reproductive system then consists of these gonads, which are the testes, and then posterior and superior to the testes, this is the epididymis. And while you produce sperm cells in the testes, these sperm cells are not mature. So the sperm cells, they travel into the epididymis to be matured, and then the epididymis continues with this long tubule that we call the vas deferens or ductus deferens. And then this tube will transfer the mature sperm cells from the epididymis into this enlargement of the ductus deferens that we call ampulla of ductus deferens. From there, the sperm cells will be combined with fluids coming from these accessory glands that we call the seminal vesicles and the prostate gland. The prostate gland surrounds the the prostatic urethra that it is found in here, and the seminal vesicles are located just posterior to the urinary bladder. Now, when you combine the fluid from the seminal vesicles with the prostate gland fluids and the sperm cells, you form a liquid that is called the semen. 90% of this fluid of the semen, it is coming from the seminal vesicles and the prostate gland. So 10% will be sperm cells. And then just below to the prostate gland, you have a couple of small glands that are called Cooper glands or bulbourethral glands. These are next to this region of the penis that is called the root of the penis. And then the penis consists of two erectile tissue cylinders on the dorsum of the penis, which is this, that are called the corpora cavernosa. If you see this name in here, corpus cavernosum is for singular, but these are two cylindrical structures. So these are filled with this tissue that it is very open, that it looks like caverns, so that's why it's called corpora cavernosa or corpus cavernosum, that during reproductive age, this erectile tissue, especially the one from the corpora cavernosa, get engorged with blood, and that leads to erection. Now, below the corpora cavernosa, we have a single cylindrical structure that is called the corpus spongiosum. The corpus spongiosum enlarges at the end of the penis, the distal end, that we call the glands of the penis. And then within the corpus spongiosum, we have the last portion of the urethra that is called the spongy urethra. The second portion of the urethra is called the membranous urethra that runs here at these muscles of the pelvic floor. Now, the body of the penis is located outside of this region. Okay, and then the glans of the penis, which is the tip of the penis, is covered by this fold of skin that it is called prepuce. And this is the one that is removed during circumcision. And underneath this prepuce, there are some glands that secrete oily substances to lubricate uh, the glands of the penis. And this skin is retractile so that the males can have washed this part so to prevent accumulation of this oily secretion of these glands that are located within the glands of the penis. And then the testicles are suspended within this skin fold that is called the scrotum. Okay, so external genitalia in the males is made by the scrotum and the penis, and they are located within the perineum, this diamond-shaped area between the thighs, and this perineum anteriorly has the pubic symphysis, laterally will be the ischial tuberosities, and posteriorly the coccyx. So this is the perineal area. It's diamond shaped, and this perineal area is subdivided into the urogenital triangle and then into the anal triangle, where you have the anus. Now within this urogenital triangle, you can see this folding that is called the perineal raphe, which points where the right and left part of the body fuses. And then here is the scrotal raphe, which is continuation of the perineal raphe at some point. So details about the scrotum. So it's a folding or a patch of skin, and it has a muscular layer and several layers of connective tissue that helps to protect the testis. Now, the left testicle, when it descends lower than the right, so they are not at the same level, so they don't compress against each other. The skin of the scrotum has sebaceous glands, sparse hair, and it is rich in sensory innervation. And it has dark pigmentation with regards to the surrounding skin. And inside this scrotum has this division or this connective tissue that separates the right and left area of the scrotum, and this is called the median septum. The perineal raphe or the scrotal raphe will be the external surface of this median septum. The spermatic cord is a tubule made out of fibrous connective tissue that has the ductus deferens, the blood vessels, the lymphatic vessels, and the testicular nerves. And this spermatic cord will run through this inguinal canal, and it helps to have this transfer of these structures so it can run between the pelvic cavity and the scrotum, these structures that can bring the sperm and also the nourishment for the testicular tissue. Again, the testes, in order to produce the sperm cells, they have to have a temperature lower than the body temperature, which is 37 degrees Celsius, and spermatogenesis can happen usually 1 degree Celsius less, 30, 36, 35, take or give. But it has to be for sure lower than 37 degrees. So in order to regulate the temperature for spermatogenesis, the scrotum has different structures. It has this skeletal muscle coming from the internal abdominal oblique muscle, which is called the cremaster. And then when the cremaster contracts, it will pull the testicles towards the body during cold temperatures, so you can have more temperatures for spermatogenesis. Now, if the weather is warm, these fibers of the oblique muscle, the cremaster muscle, will relax, so that helps to descend the testis. Along with this scrotum, there is this smooth muscle layer that is located within the skin of the scrotum that is called the dartos muscle or dartos fascia. This will contract when it's cold, this dartos fascia, making the scrotum to look wrinkled. And then when the body temperature is warm, the dartos fascia will relax, so it will unwrinkle the scrotum so that you have more heat loss. So these are the layers of the spermatic cord and the scrotum. So here within the spermatic cord, you have this cremaster muscle, which is part of the internal oblique, the external oblique strips, sorry, internal oblique strips. So this is the cremaster muscle, which is protecting the structures inside the spermatic cord, which are the veins in this area that are called the pampiniform plexus, and also the testicular artery and the testicular nerves, and also the lymphatics. And then outside of the cremaster muscle, there is this fascia of the spermatic cord. Now, here in the scrotum, we have this fascia that it is called the tunica vaginalis first, and then you have this cremaster muscle in here that you can see better in this side, and then you have this dartos muscle in here, or dartos fascia, and then finally you have the skin of the scrotum, and then in here you have the median septum that separates the right compartment from the left compartment. So what is the pampiniform plexus? Is a network of veins that interconnect, that comes from the testis to drain the blood from the testis and surrounds the testicular artery and the spermatic cord. Because of this close connection between the artery and the veins, you can have exchange of heat that helps to cool the temperature, so that when you are transferring the sperm cells along this ductus deferens, the sperm cells that are mature, they still being transferred at a lower temperature than the body temperature, so that you maintain viability of these sperm cells. So this is how this countercurrent exchange mechanism for heat happens. So in here you have the testicular artery that is bringing blood from the pelvic cavity into the testis. And then in here on this side, you have the pampiniform plexus with a series of veins that are interconnected that drains the blood from the testis into the blood vessels within the pelvic cavity. As the blood is coming in different directions, so the blood that it was found in the pelvic cavity at 37 degrees Celsius, you have transfer of this heat from the blood into the veins, so that the veins help to cool off the blood, so that when the blood reaches these testicles, you'll have 35 degrees Celsius, and then you have optimal temperature for developing of the sperm cells. But at the same point, as you're gradually decreasing the temperature, and you have somewhere in here, and it is not depicted in this diagram, you have the ductus deferens and the sperm cells that are coming off from the epididymis, going through the ductus deferens. They also have a lower temperature, so they can move without having high temperature and having any damage. So this is the countercurrent exchange of heat. Okay, so for the testes. So the testes are the gamete-producing organs of the males. They are located outside the body in the scrotum. And they are considered mixed glands. So they are endocrine because they secrete testosterone and inhibin that goes into the bloodstream. And they are exocrine because they secrete the sperm cells to the outside of the body. The diameter or the dimensions of the testes are 4 by 2.5 centimeters. They have an oval shape that is slightly flattened, and they are covered by this membrane that is called the tunica vaginalis. And inside the tunica vaginalis, there is this other membrane or a connective tissue that is called the tunica albuginea. And this tunica albuginea will deepen into the testes, separating the testes into lobules. And this actually tunica albuginea, as it's migrating into the deeper portion of the testes, it will form this septum. And then you will have between 250 to 300 wedge-shaped lobules. Inside these lobules, there are tiny tubules that are called the seminiferous tubules, which are the tubules where the sperm cells are being made. So inside these lobules of the sperm cells, you have between 2 to 3 seminiferous tubules. And then within these walls of these seminiferous tubules, you have cells at different levels of differentiation. The outer cells are the germinal cells or germ cells, and these germ cells are called spermatogonia. And surrounding this spermatogonia, you have the nerve cells. So the nerve cells are located between the germ cells, and they help to protect the germ cells and promote their development. Along with that, these nerve cells will produce these nutrients for the germ cells and also for the cells that derive from the germ cells. So the different levels of spermatocytes are as they are developing. And then these nerve cells also will remove the wastes produced by the germ cells. They provide growth factors, and they will form what it is called the blood-testis barrier. So the sperm cells, as they're developing, they're different from the DNA of our rest of our cells. And our immune system, or the immune system, will detect those differences in the genetic material, and it will tend to attack them. So these nerve cells, because they are in close contact with these germ cells, they form the blood-testis barrier so that the immune cells won't attack the sperm cells. Now, within the seminiferous tubules, we also have a different type of cells that are called the interstitial cells. And these interstitial cells are endocrine cells that produce testosterone. And this testosterone will act as a growth factor so that the sperm cell development can happen. So interstitial cells are located between the seminiferous tubules, and they also respond to the action of luteinizing hormone. This was part of the key points that I told you, that the interstitial cells are located between the seminiferous tubules. They are cells that respond to the action of luteinizing hormone, and they are also known as Leydig cells, Leydig, and they produce testosterone. Now, the nerve cells, the other key point, again, they are located inside the seminiferous tubules. They coordinate spermatogenesis, they form the blood-testis barrier, and they secrete inhibin hormone. So that was the other key point. So this is light microscopy at high magnification of the seminiferous tubules and the interstitial cells. So this is the base of a seminiferous tubules. In here you will have the spermatogonia just at this area. And then as the spermatogonia is dividing towards the lumen of the seminiferous tubule, which is this. And if you see here, you see a lot of lines there. These are the sperm cells, the spermatocytes. So from differentiation levels, the most immature cells will be here at this first row of cells. Then these cells will divide the spermatogonia, will form primary spermatocytes and then secondary spermatocytes. And then these will be more mature, and then they will keep maturing more until they reach into the lumen of the seminiferous tubules. And then in here, we will have in this area the nerve cells that form the blood-testis barrier, and they provide nourishment for these developing cells. And outside of these, we have the interstitial cells that has the different functions that I already told you about. So here you can see primary spermatocytes developing, and then in here you have secondary spermatocytes, and you will have a spermatozoa here and spermatids in here. So this name, spermatogonium or spermatogonia, will be the most immature cells. So the blood-testis barrier are the tight junctions that happens between the nerve cells. So it separates the sperm cells from the immune system, preventing antibodies or other large molecules affecting the spermatogonia, so that you don't have this spermatogonia attack, or attack into the spermatogonia, I should say. So again, this is the light microscopy of the sperm cell, seminiferous tubules, spermatogonia in here, the first row, and then primary, secondary spermatocytes, and then spermatids, and then a spermatozoa in here. And then nerve cells in the blood-testis barrier, and then you have here the interstitial cells. Okay, so when you produce these sperm cells within the seminiferous tubules inside the lobules of the testes, they will migrate into an area that it is superior within these lobules inside the testes, that is called the rete testis. So the rete testis will be the network that collects the sperm cells from the seminiferous tubules. And then from there, the sperm cells will go from the rete testis into the epididymis, so they can mature. Blood supply into the testes happens by the testicular artery, which is a branch of the aorta, and the testicular artery has low blood pressure, and also provides this blood supply to the testes. And within the sperm cells, you have large mitochondria because you have poor oxygen supply, and you will produce more oxygen in the mitochondria to help the sperm cells to survive from the hypoxic environment within the female reproductive tract. Drainage of the blood from testes into the pampiniform plexus that runs inside the spermatic cord. And then from the pampiniform plexus, the blood from the right testicular vein will drain into the inferior vena cava, while the left is going to drain into the left renal vein. Innervation to the testes happens by T10 and T11 spinal nerves, and they will carry sensory fibers that delivers pain sensation through the sympathetic system, and also regulates blood flow. This is a sagittal section of part of the testicles. So you can see the tunica vaginalis here, and then the tunica albuginea here. The tunica albuginea will deepen into the testes, forming these lobules, these wedge-shaped structures. And then inside the lobules, you have these tubules, seminiferous tubules, where you have spermatogenesis going on. And then once spermatogenesis happens, these sperm cells are going to be transferred into the retetestis in here. And from the retetestis, the sperm cells will migrate into the epididymis, which is a dolphin-like structure that it surrounds the superior pole of the testis, which is this, and the posterior pole of the testis. The epididymis is the organ for maturation of the sperm cells. So it has a head, a body, and a tail. And then once the sperm is mature, it will get transferred from the tail of the epididymis into this vas deferens or ductus deferens. And then it will go through the spermatic cord. So spermatic ducts, they run from the testis into the urethra, into the prostatic urethra. So these are efferent ductules that delivers this sperm from the epididymis into the ejaculatory duct. Now, so in the epididymis, when you have this maturation of the sperm, it will take between 40 to 60 days to happen. And the sperm, if it's not ejaculated, it's going to be reabsorbed within the epididymis and nothing happens. The vas deferens or ductus deferens, it's a muscular tube that has a length of 45 centimeters. It will go from the epididymis, and then it will go into this spermatic cord, traveling through the inguinal canal, and it will surround the posterior superior portion of the bladder, and it will reach this ampulla of the ductus deferens, which is a widened part. And this ampulla of the ductus deferens is going to join the duct of the seminal vesicle, and they will form a small area that it measures 2 centimeters in diameter that is called the ejaculatory duct. And then this ejaculatory duct will receive the fluid coming from the prostate gland and also the seminal vesicles. This is a cross section of the ductus deferens, so you can see the different layers. So you have this inner layer that it is made of an epithelium. And then you have this muscular layer, an inner longitudinal layer, and a middle circular layer, and another longitudinal layer. These layers are innervated by the sympathetic nerves so that you can have peristalsis during emission of the sperm cells. And then you have another layer that it is made of the connective tissue or adventitia. So this is the ductus deferens or vas deferens. So how it goes from the scrotum, passes through the inguinal canal, and then it goes and surrounds superiorly and a little bit posteriorly the urinary bladder, and then widens within the ampulla. And then the ampulla will join this duct of the seminal vesicle, and together they will form the ejaculatory duct. So that you can have the sperm cells receiving the fluid from the seminal vesicle and then the fluid from the prostate gland, and you form semen. So semen is 90% fluid coming from the seminal vesicles, the prostate gland, and 10% sperm cells. This is a longitudinal cut of the ductus deferens and how it widens in this area, the ampulla of the ductus deferens, and see how it joins this duct of the seminal vesicle. So this is the seminal vesicle. It's a lobular gland that it is found at the base of the urinary bladder, which is this. And then you have the ejaculatory duct. So if you want to remember the trajectory of the sperm cells through the male reproductive organs through to the exit of the body, you can remember the acronym 7UP. So the sperm cells travel from the seminiferous tubules into the epididymis, into the vas deferens, into the ejaculatory duct, and then into nothing, and then into the urethra, prostatic urethra, and then into the penis. 7UP. So the correct order then, that's one way to remember. So S, E, B, E, N, P, U, and P. About the accessory glands, so we have three. We have seminal vesicles, prostate gland, and bulbourethral glands, or also known as Kupfer glands, C-O-W-P-E-R. The seminal vesicles, they are more active in terms of producing fluid. The fluid at the seminal vesicles, it's rich in fructose, citric acid, and in bicarbonate ions. The purpose of having fructose within this seminal vesicle fluid is to nourish the sperm cells. As they are traveling, they have to migrate really rapidly and they move really fast. They have like a zigzag motion and they have to use a source of energy to move. So they use fructose. That's why they have a lot of mitochondria. So part of this fructose, it is converted into glucose and then it goes into the mitochondria so you can generate ATP so that the sperm cells can move fast. So the seminal vesicles are very active and they form 60% of the semen, while the prostate gland also secretes an alkaline fluid that is rich in bicarbonate, and they also produce some substances to nourish the sperm cells, but they are less active and they form 30% of the semen. And the fluid of the prostate gland is kind of thin and milky. And again, prostate gland surrounds the urethra, the prostatic urethra, while the seminal vesicles, they don't surround the urethra. They are located posterior to the urinary bladder. So before we move into the details about the bulbourethral gland, so one reason for why both the prostate gland and the seminal vesicle secretes this alkaline fluid, it is to neutralize the pH of the vagina in females, which is acidic. So it's to neutralize this pH so that the sperm cells can migrate much easier, because if not, the sperm cells will be killed by the acidity. Okay, so the bulbourethral glands, they are located next to the root of the penis, on the sides of the root of the penis, and they secrete this thick, crystal clear color fluid that lubricates the urethra before the ejaculation process so that you have a slippery lumen within the urethra so that the uh, sperm cells travel much easier through the urethra. Along with that, it will help to get rid of any acidity from the urine that it is located within the urethra. So for the penis is the copulatory organ. It deposits the semen into the female vagina, and half of the penis is located internally, so it forms the root, while half of the penis is external, so it's forming the shaft or the mid portion and the glans, the tip of the penis. The length of the penis when it's not erect is 4 inches in length and between 5 to 7 inches when it's erect. It is covered by skin, and the skin of the penis that is retractable will be the one that is located within the glans, and this one is removed during circumcision. Now, the glands that are located within the glands that are located within the glands or tip of the penis, they secrete this oily secretion that is called smegma, that at some point helps to lubricate the glands too. Internally, the penis is made by these cylindrical structures that pair corpora cavernosa and the single corpus spongiosum. The corpus spongiosum encircles the spongy or penile urethra, and then they dilate or enlarge in the glands. Now, the proximal end, or the end that it is close to the root, is encircled by the bulbospongiosum muscle, while the base of the corpora cavernosa, the one that is located at the root, is called the crus, and this is covered by the ischiocavernosus muscle, which helps to stabilize erection. The three cylinders have erectile tissue, and especially within the corpora cavernosa, you have these blood sinuses that are called the lacunae, and then you have partitions that are called the trabecula. You also have that in the spongy, but these are more prominent within the corpora cavernosa. So this is a cross section, so you can see the structure of the corpora cavernosa here in the dorsum of the penis, and this is the corpus spongiosum surrounding the urethra. And then you have the deep penile artery here in the middle of this corpora cavernosa, and then externally to this corpora cavernosa you have the dorsal artery and dorsal vein of the penis and the dorsal nerve. And then the penis is surrounded by this deep fascia, and then this superficial fascia and the skin. Now within the glands, of course, I already told you have this prepuce, but you have a frenulum in the ventral surface, so that you can have this prepuce, just in case that the male is not circumcised, still attached to the base of the glands, preventing this folding of skin from unfolding, basically. And then you have at the tip of the glands the external urethral orifice. Okay, so production of gametes in both males and females happens through this special process of cell division that is called meiosis. So you have two meiosis steps, which is meiosis 1 or first division, and meiosis 2 or second division. It's basically similar to mitosis, in which you have prophase, metaphase, anaphase, and telophase. But in meiosis 1, you have genetic exchange in prophase. In mitosis, you never have genetic exchange. You always produce daughter cells, hopefully with the same genetic material. In meiosis, genetic diversity is responsible for uh, the different traits that we see in humans. So no single human is identical to other, 100% identical. We are very alike to some of our siblings or our parents or grandparents or family members, but never identical. And this is because during gametogenesis or formation of gametes, you have genetic exchange in prophase. And this is how it happens. So during early prophase, you have condensation of the chromatin similar to mitosis, in which you have the DNA condensing, forming visible chromosomes. And then you have these chromatids of the chromosomes joining in the center, which is called the centromere. As prophase is happening, you'll have crossing of some of these sister chromatids into what we call the crossing over, and then you will have exchange or swapping of pieces of the parental chromosome. So you have chromosomes from mom and dad side, and you have this exchange or crossing over, and then you form a newly set of chromosomes. The actual physical exchange happens then of these segments of genes, and this other name for crossing over is called chiasmata. Now, after this crossing over happens in prophase 1 of meiosis 1, you then align the chromosomes in the equator or the center of the cells into what we call metaphase plate in metaphase 1. And then as you are forming this alignment of the chromosomes, you will create microtubules that you will bind into the centromeres. And then in anaphase 1, this shortening of these microtubules will pull these chromosomes, homologous chromosomes, into two different cells. And then in telophase, the last part of the meiosis 1, you will have cytokinesis, in which you have a division of the cytoplasm, and also you have at the same time the actual separated set of chromosomes surrounded by a new nuclear membrane. And then in here you have two daughter cells that will be ready to start meiosis 2. In meiosis 2, you have again prophase, metaphase, anaphase, telophase, which will be now called prophase 2, metaphase 2, anaphase 2, and telophase 2. And you start dividing the number of chromosomes that you started with in here, which was 46, into two sets of cells for each of these daughter cells, which will have 23 chromosomes each. So you started with diploid cells in here, or cells that has 46 chromosomes, and then you end up with cells that has 23 chromosomes, which are known as haploid cells. So we have two types of cell division. We have mitosis and we have meiosis. In mitosis, the body cell doubles the DNA. So you have DNA duplication, and then you divide this duplicated DNA to give your two daughter cells identical number of chromosomes. So you don't change this number of chromosomes. So our diploid cells will have 46 chromosomes or 23 pairs. When you duplicate this number, if you multiply 46 times 2, that will be 92. And then if you divide these 92 chromosomes into two daughter cells, you have to give 46 each. And then in here, you don't have genetic exchange. And then you undergo, in order to have mitosis, these stages: prophase, metaphase, anaphase, and telophase. And you don't have 1 and 2. It's just purely prophase, metaphase, anaphase, and telophase. In meiosis, you undergo this cell division twice, so those diploid cells end up having half of the number of chromosomes. So from 46, you end up with 23 chromosomes. This is called haploid cells. Meiosis is also known as reduction division. And through meiosis, you have genetic diversity because you exchange chromosomes from the parents. I already explained you. So this. So after meiosis 1, each cell has 23 chromosomes, but each chromosome is double-stranded. So these chromosomes has these chromatids, sister chromatids attached to each other. And then during meiosis 2, these chromosomes divide, so they will have 23 single-stranded chromosomes, while in meiosis 1 you have 23 double-stranded chromosomes. So you separate the sister chromatids. So the actual production of sperm cells is spermatogenesis that happens in the seminiferous tubules. And as this spermatogenesis is happening, you have this remodeling of these spermatogonia that started as large cells into small mobile cells that we call the spermatozoa. And then you reduce, as you are doing this, the chromosome number in half, and then you start also reducing the volume of the cytoplasm. And again, in meiosis, you recombine the genes, and then you produce, out of one spermatogonia, 4 daughter cells that will have 23 chromosomes each, single-stranded chromosomes. Semen is seminal fluid that it is expelled during orgasm, and then you have this volume of semen, which can range between 2 to 5 milliliters, and 60% of the seminal, of the fluid is, or the semen, is going to be coming from seminal vesicles, 30% from the prostate gland, and 10% from the sperm and other spermatic duct secretions. The normal count of sperm cells is between 50 to 120 million cells per milliliter. So if you want to calculate the total, you just multiply times 5 for the maximum, and then the lower count will be between 20 to 25%, which basically is suggestive of infertility. Contents of the prostatic fluid. So it has calcium citrate, phosphate ions, and a clothing enzyme. And also has this prostatic specific antigen that it is a serine protease. And the seminal vesicles, the fluid is viscous, yellowish, and it has fructose, other carbohydrates, it has citric acid or citrate, and prostaglandins, and a protein called proseminogyllin. The semen is going to be thick and it's going to be sticky. This promotes fertilization so that the sperm cell has basically this surface to migrate towards the female reproductive tract, and it has this clothing enzyme from the prostate gland that activates this protein from the seminal vesicles that is called proseminogyllin, that it will convert into a fibrin-like protein that is called seminogyllin that helps to entangle the sperm, and this helps to stick the sperm into the inner wall of the vagina and the cervix. This will ensure that the semen does not drain back into the vagina, and it will promote the sperm to reach the uterus. So it between 20 to 30 minutes after ejaculation, the serum protease, the prostatic antigen fluid, will break down the seminal gelin and liquefies the semen, releasing the sperm cells. So the requirements for sperm motility will be having an elevated pH and a high energy source. Prostatic fluid will buffer the vaginal acidity, which is between 3.5 to 7.5, and then the seminal vesicles will provide the fructose so that you have production of ATP in the mitochondria of the sperm. And then the sperm will be ready to crawl up the vagina, the uterus, the uterine tubes, and reach the oocyte. Now, another function of the fluid, the semen, is that it's reaching prostaglandins, that is going to thin the mucus at the cervical canal and also stimulate contraction of the uterus and the uterine tubes to help to spread the semen. So this is the diagram showing you spermatogenesis. So you have the oogonia with 2N or duplicated chromosomes that will migrate from the basement membrane of the seminiferous tubule up towards the lumen and forming type 2 spermatogonium. So type 1 will be the original spermatogonium, and then it will be converted into type B spermatogonium, or a secondary, another type of spermatogonium. So from there, the type B spermatogonium will have changes into what we call the primary spermatocyte. So you will have a smaller nucleus and a smaller number of cytoplasm. So you have structural changes. So you have a primary spermatocyte which will enter meiosis 2, and it will be converted into 2 secondary spermatocytes, now with half of the number of DNA. So instead of 2N, it's going to become N. And then these secondary spermatocytes will enter into meiosis 2, and it will form these 4 cells or 4 spermatids that will have 23 single-stranded chromosomes, while this one has 23 double-stranded chromosomes. And then from there, these spermatids will migrate toward the lumen of these seminiferous tubules, and through a series of structural changes that takes about a week, they will form an elongated cell that is going to be called the spermatozoa. The transformation of these round cells into these cells that looks like mature sperm cells is called spermiogenesis. So the actual spermatogonia form in the yolk sac in the embryo, and then they migrate to the sperm cell, to the testicles, and they colonize the testicles, and they become spermatogonia. And then under the influence of the testosterone that is released during puberty, you will have the activation of mitosis of this spermatogonia, so you can form this spermatozoa and later on these sperm cells. So spermiogenesis is the formation of 4 spermatids, now with the structural changes that makes them look like the actual mature sperm cells, and then you will migrate these immature sperm cells into the epididymis, so you can form a mature sperm cells. So during spermatogenesis, you have migration then of these cells towards the lumen, and then from the lumen they will migrate into the epididymis. So this is a process of spermatogenesis that includes the change of this spermatogonia into these secondary spermatocytes and then into these spermatids, in which you have the nucleus decreasing its size, and then you have the formation of a structure next to the nucleus that is called the acrosomal vesicles. And these acrosomal vesicles, they have enzymes that will help to disrupt the surrounding cells of the oocyte so that the fertilization process can happen. Along with this, you start having the formation of more and more mitochondria and then the formation of flagella. So these flagella will be a long extension of the sperm cells that helps to sweep this structure sideways so that the sperm cell can undergo motion towards the reproductive tract of the females. The actual process of spermatogenesis will involve the change of this circular form of cells into kind of a diamond-shaped head that has a large nucleus, very little cytoplasm, the acrosome at the tip, and then you will have a neck in here that will be connected to this mid piece of the tail that is where you have the mitochondria that is going to produce the ATP so that the flagella can move, and then you can have the transferring of the sperm cells. The spermatozoon will be the name of the cell, the mature sperm cell that it has a head and a tail. The head is pear-shaped or diamond-shaped, and in here you have again the nucleus, the acrosome with the acrosomal enzymes, and then it will be connected to the basal body, which is the area at the end of the nucleus where the flagellum attaches. The tail has the mid piece where you have the mitochondria, the main piece that it has a membrane that is called the axoneme, and then you have the end piece, which is the tip of the flagella. And this is the actual mature spermatozoon. Again, the diamond-shaped head with very little cytoplasm. It has this acrosome filled with enzymes, and then it's connected to this basal body, which is connected to the mid piece that it is filled with the mitochondria, and then you have the main piece and the end tail of the flagella. And inside this flagella, there is many proteins inside this axoneme, which helps in the motility of the sperm cells. Okay, so that's all for the male reproductive tract. Now for the female reproductive tract. So again, just as a reminder, so between weeks 8 to 10, you cannot distinguish in utero whether it's a female or a male. So the female reproductive tract will develop from an area of the embryo that is called the paramesonephric ducts. And in the absence of testosterone and a Mullerian inhibiting factor, these paramesonephric ducts will form into the female reproductive tract. So if you don't have testosterone, you have the mesonephric ducts, the actual part that develops into the male reproductive tract, to degenerate. And then the genital tubercle, the one that is going to become the penis in males, will become the glands in the clitoris of females. And then the urogenital folds will form the labia minora, while the labioscrotal folds will form the labia majora. And then without the Mullerian inhibiting factor, the paramesonephric ducts will develop into the oviducts or uterine tubes and into the uterus and vagina. The female reproductive tract consists of internal genitalia like the ovaries, the uterine tubes, the uterus, and the vagina. External genitalia will be clitoris, labia minora, and labia majora that are on the sides of an area of the female reproductive tract that is called the vestibule, and then they are located in the mid region of the thigh that is called the perineum. And then again, primary sex organs, which are the gonads, the ones that produces the gametes and the hormones, are in the female the ovaries. Secondary sex organs will be both internal and external genitalia. This is the mid-sagittal cut of the female genitalia. So you can see here the uterine tubes. This is the uterus. This is the ovaries. This is the vagina. All of these are internal genitalia. External genitalia will be a folding of skin that is hairless. This is the labia minora or labius majus for singular, labia majora or labius majus majus for singular, and then here is the clitoris, and this area is the vestibule. The ovaries have a function to produce sex hormones like estrogens, progesterone, and also produce the reproductive cells, the gametes or ova. They have a shape of an almond. They are located within the pelvic cavity in a region that we call the ovarian fossa, that is located towards the posterior lateral part of the pelvic cavity. And the ovaries are surrounded by a membrane that's called the tunica albuginea, similar to the tunica albuginea that surrounds the testes. It has two major regions. The outer region that is known as cortex, where the actual ova develop into what we call follicles, and it has an internal region that is called the medulla, into which the blood vessels come in and out, and the nerves and the lymphatics come in and out. Ovulation is the process of releasing an egg cell, and the eggs cells develop in a fluid filled cavity that is called the follicles. The ovaries are held into their position by ligaments. So we have ovarian ligaments that attaches the ovaries into the sides of the uterus. And then we have these ligaments that are called the suspensory ligaments that holds the ovaries into the pelvic cavity. And within these suspensory ligaments, you have also the ovarian blood vessels and the nerves. And these ligaments are held into a major ligament that is called the broad ligament, that it's having a subdivision that protects the ovary that is called the mesovarium. Blood supply to the ovary is given by the ovarian artery, that it is the equivalent to the testicular artery, that it is a branch of the aorta. Now, the uterine arteries are branches of the iliac, the internal iliac artery. And both the ovarian and uterine arteries anastomose along the margin of the ovary, and then they give multiple small branches that enters the ovary. The ovarian veins, lymphatics, and nerves also travels within the suspensory ligament, and the ovarian veins, they drain into the renal vein in the left side or into the inferior vena cava on the left side. So this is the cross section of the ovaries or longitudinal section, but what you can see here are the two major regions. So this outer region is called the cortex. This is where you have the development of the egg cells, and then this region, it is the medulla, where the ovarian blood vessels come in and out, the nerves comes in, the lymphatics comes in and out. So things that you can see here in the cortex are the actual equivalent to the spermatogonia in males, which for us is called oogonia or primordial follicles. So these will be the stem cells that are going to make the mature egg cells. And then these primordial follicles, under the influence of hormones during puberty, start undergoing structural and chemical changes that will form primary follicles, secondary follicles, tertiary follicles, and a mature follicle that ultimately is going to release this oocyte in the middle of the menstrual cycle. And then once you ovulate this oocyte, this remnant will form what is called the corpus luteum, that is going to secrete progesterone. And then if no fertilization happens in pregnancy, then this corpus luteum will become the corpus albicans. And this is the capsule of the ovary. This is a microscopy of a secondary oocyte. The secondary oocytes are located here within this mid region. They are cells that are round cells that are surrounded by a structure is called corona radiata. This is the part that the sperm cells are going to penetrate through this enzymatic reaction that is going to open the corona radiata so that this oocyte can be fertilized. And then the secondary oocyte will be surrounded by this fluid that is located within this space that we call the antrum. For the uterine tubes, they are also known as oviduct or fallopian tubes. They have a length of 10 centimeters of diameter, and they are connected to the superior lateral portion of the uterus. They are a muscular tube that it is lined with cells, cuboidal cells that has cilia, and they are highly folded. Now, the lumen of the uterine tubes has very thin diameter. It's almost like the the size of the a human hair, the diameter of a human hair. Now, the fallopian tubes, they have a distal end that it is called the infundibulum. It's a funnel-like extension. And this funnel-like extension ends in these finger-like projections that we call the fimbriae. When an egg cell is ovulated, the fimbriae start doing this contraction and relaxation as to attract the oocyte. The oocyte are actually ovulated into the peritoneal cavity, so they need to be transferred into the uterine tubes. And there is no direct physical connection between the fimbriae and the ovaries. So that's why sometimes fertilization can happen within the peritoneal cavity in what we call ectopic pregnancy. And ectopic pregnancy also can happen when you have an obstruction of any of these parts of the fallopian tube. So the fertilized egg can start growing within these regions of the fallopian tubes, but because you have a narrowing of this or an obstruction within these areas of the fallopian tubes, the fertilized egg cannot travel into the uterus, and then you have enlargement of the fallopian tube and then rupture and hemorrhage, which leads to basically sometimes death. So again, this is the infundibulum, and then you have the ampulla, and then you have the isthmus. Fertilization happens between the boundary of the isthmus and the ampulla. And the isthmus is a constricted region that connects into the superior lateral portion of the uterus, which is this. And then here we have the ovaries. suspended by this ovarian ligament to the sides of the uterus. And this is part of the mesosalpinx, part of the broad ligament that helps and protects this uterine tube from moving up and down or laterally. So here is the suspensory ligament holding the ovaries to the lateral posterior wall of the pelvic cavity, bringing the ovarian veins and the lymphatics and the nerves into the medulla of the ovaries. And then the mesovarium will be the part of this broad ligament that protects the ovaries. So I already talked about these portions of the uterine tubes and the mesosalpinx, which is the part of the broad ligament that enfolds the uterine tube. This is the scan electron micrography of the epithelium of the uterine tube. So these are cuboidal cells, and these cuboidal cells have cilia. This cilia sweeps so it can bring the fertilized or unfertilized egg into the uterus. When someone has an STD, the bacteria starts destroying these cells and these cilia, and then it creates scars that can obstruct the lumen of the uterine tubes. For the uterus, it's the organ that harbors the fetus, and it has an inverted pear shape that has different regions. It has a dome shape upper region that is called the fundus, a mid portion that is called the body of the or the corpus, and then it has this extension coming off from the body that enters into the vagina that is called the cervix. The normal position of the uterus is anteroversion or tilting forward over the bladder. It shouldn't be tilting so much forward that it will compress the urinary bladder. The lumen of the uterus is triangular, and it has two openings to the uterine tubes, and it has a lower part that is called the internal os, and it's not a hollow cavity, it's a potential space for just in case that the female becomes pregnant. Now, within the lower part of the uterus, we have the cervical canal that connects the lumen of the uterus to the vagina. And within this cervical canal, we have the internal os or superior opening, and then the external os or inferior opening that connects to the vagina. Within the cervix, the uterus has cervical glands that secretes this mucus that prevents the spreading of the microorganisms from the vagina to the uterus. So this is the anterior view of the uterus. So this is the fundus, this is the body, and this is the cervix. This is the lumen, and then in here you have the internal os and the external os and the cervical canal between the two of them. And the cervix is made out of a wall that has this epithelium that is known as endometrium, and we have functional endometrium, and we have this endometrium falling off during reproductive stage every month into what we call menses. And then the next layer is called the myometrium. It's the thickest layer. It's made of smooth muscle that responds to hormones during the menstrual cycle together with the endometrium. And also we have the last portion that is called the perimetrium. The uterus is also protected by the broad ligament, and we have then these several ligaments and structures that runs within the broad ligament. So we have the round ligament that actually runs from the lateral portion superolateral portion of the uterus into the ischial tuberosity, passing through the inguinal canal. And then we have this cardinal ligament that helps to actually not only uh, hold the inferior portion of the uterus, but also part of the vagina into the sides of the pelvic cavity. And then we have posteriorly this uterosacral ligament that prevents the uterus to fold over so much over the anterior urinary bladder. Now, when some females, not in all, but after several pregnancies, many females suffer urinary incontinence, or they have to go and pee a lot. It's because these uterosacral ligaments elongate so much, and then they don't fold back into their normal length. In order to prevent this, then or to prevent the female from having accidents uh, as they are laughing or coughing and having a little bit of urination, these uterosacral ligaments can be shortened during surgery and then pulled back posteriorly to be the uterus. So again, the regions of the uterine wall will be perimetrium externally, myometrium medially, and then endometrium. During pregnancy, close to weeks 39, 38, this myometrium undergoes contraction to expel the fetus, and this will these smooth muscle fibers respond to oxytocin levels. Endometrium is simple columnar epithelium, and within the endometrium we have also tubular glands that secretes nutrient like glycogen to help nourish the embryo just in case that the female is pregnant. It has this functional layer that it is shed every menstrual cycle during reproductive life or stage, and then it has this basal layer or stratum basalis which renews this functional layer. So this is the microscopy of the uterine wall. What you can see here in this base portion will be the myometrium, and then you have here the stratum basale of the endometrium, and then this will be the functional layer, or the endometrium that falls off every end of the menstrual cycle. And then you have here these glands that secretes glycogen just in case that there is fertilization. So this is the simple columnar epithelium. These are the endometrial glands, and this is the lamina propria. So I already talked about these ligaments. Another name for the cardinal ligament is lateral cervical ligament, and I already told you how they connect. Blood supply to the uterus is given by the uterine artery, which is part of the internal iliac artery, and also by branches of the ovarian artery, which is a branch of the aorta. And they anastomose the uterine artery branches together with the ovarian arteries. They run on the body of the uterus outside, and then they penetrate the walls of the uterus. So they go inside, they penetrate, and they form what we call the spiral arteries. And then these spiral arteries will undergo also cyclical changes during reproductive stage. So they will start elongating towards the surface of the endometrium, and then with the influence of hormones, they will rhythmically constrict and dilate, making the mucosa alternatively blanch and flush with blood. Especially, this will happen during the menses. And what it's going to do when you have this constriction is that you will have less oxygen going into the endometrium, and then these hypoxic conditions will lead to the shed of the endometrial cells that are hypoxic, and this constriction will lead to the cramps that we feel during the menstrual cycle. So here is the blood supply then to the female reproductive tract. So we have this ovarian artery, branch of the aorta, and this uterine artery, branch of the iliac artery. This runs through here, the ovarian artery through the suspensory ligament, providing blood supply to the ovaries, to the fallopian tubes, and then part of the superior part of the body of the uterus that is going to anastomose with branches of the uterine arteries in here. And then both of them, they penetrate, forming first the arcuate arteries and then the spiral arteries deeper. That goes and provides blood supply to the endometrium. Now for the vagina. The vagina, it's a hollow organ, muscular organ that measures between 8 to 10 centimeters in diameter, or sorry, in length. And it helps for the discharge of the menstrual fluid, is the copulatory organ to receive the penis and the semen, and it's part of the birth canal. It has different layers. So the internal layer is mucosa, so it's a stratified squamous epithelium without keratin, and the next layer will be the muscular layer, and then outer to this muscular layer you have an adventitia. It is tilted posteriorly between the rectum and the urethra, and the vagina has no glands. Superiorly, in between the cervix and the lateral wall of the vagina, there are some blind-ended spaces that we call the fornices. And then the walls of the vagina, they have rugate, transverse ridges, that helps for basically during the copulation to unfold and enlarge the vagina so it can allows the entrance of the entire penis. Now at the end of the vagina in virgins there is this mucosal fold that is called the hymen. So before reproductive stage, the vagina has not stratified squamous epithelium, but it has simple cuboidal epithelium. So during puberty, when estrogen levels are high, this epithelium will become stratified squamous. Why stratified squamous? Because it's a type of epithelium that it is more protective than simple cuboidal. And during reproductive stage, penetration of the penis, if the female is sexually active, it will require this epithelium that it is stronger. Now, inside the vagina, there are bacteria that is part of the normal flora that will ferment glycogen, producing acids that will drop the vagina pH, that inhibits the growth of bacteria that is harmful that we call pathogens. And also within the vagina, there are antigen presenting cells that are called dendritic cells that can be infected by HIV. So this is the microscopy of the vagina. So this will be the muscularis layer, and this will be stratified squamous epithelium. Why is this? Well, because this is a vagina sample from a female in reproductive stage. Okay, so for the external genitalia, it is the name for a series of structures that are located within the vulva or pudendum. So we have this structure within the anterior wall in the pubic area of our body that's called the mons pubis. It's a amount of fat over the pubic symphysis that has pubic hair. Why is the reason for this? We don't like it. Many of us, it looks not aesthetic, if you want to call it that way. Well, it's for protection. What do we have behind the pubic bone? We have the uterus. So we have this fat of pad there for cushioning. Now, then below in the actual perineal area, we have the labia majora, which is a fall of skin. Is very thick, and it is filled inside this fall of skin with adipose tissue, and this has hair in females that are after reproductive stage, that are after puberty. So that are in this stage of life. And then inside the labia majora, there is this fold of skin that is thin skin that is hairless that is called the labia minora. And then on the sides of the labia minora, there is an opening for the vestibular glands that are called also Bartolini glands. That these glands helps to secrete this fluid that is for lubrication during sexual intercourse. So the clitoris will be anterior to the vulva or into the vestibule, the area in between the labia minora, and this is an erectile organ similar basically to the male penis. It has a glans, it has a body, it has a crura. So basically at some point it has similar regions as the male penis. And then again, within the sides of this vestibule, we have the greater vestibular glands, but also we have minor glands that are called the lesser vestibular glands or the periurethral glands that again secretes this fluid that helps to lubricate the male the female vestibule. So this is the female perineum. So we have the mons pubis here. Here we have the prepuce of the clitoris. We have the clitoris in here. And then we have the external urethral orifice in here. Sorry, it's in here. And then here is the labia minora and labia majora. And this space is called the vestibule. And then here is the vaginal orifice. And in here we have the openings for the vestibular glands. So here is the location of the vestibular glands. So if you remove this skin in here and these structures, you can see them. So these are the greater vestibular glands. And then these are the paraurethral glands, and the lesser are in here. Now let's move into oogenesis and the sexual cycle. So in general, the function of the reproductive organs, whether they are from the male or the female, is for perpetuation of species or transfer of genetic information. So we undergo then these cyclic changes, especially in females, for producing these egg cells that is going to help for the functioning of the reproductive tract, reproductive system. So the reproductive cycle can be defined as a sequence of events from fertilization to giving birth and returning to fertility. And the sexual cycle will be the events that recur every month when pregnancy does not intervene. So this sexual cycle consists of two interrelated cycles, the ovarian cycle and the menstrual cycle. Ovarian cycle happens, of course, in the ovaries, and menstrual cycle happens in the uterus. Oogenesis is the production of egg cells, and during oogenesis, diploid cells, the oogonia, forms haploid cells or egg cells. Now, in order to produce these haploid cells, you will have cyclic changes in hormone secretion. Embryonic development of the ovaries. So, the oogonia develop from the yolk sac in the embryo, similarly as to males. And then this oogonia will colonize the gonadal ridges during the first 6 weeks of development, and then they will differentiate into oogonia, and then they will start multiplying until the 5th month, and we will end up with 6 to 7 oogonial cells. Now, these oogonial cells will form primary oocytes in meiosis 1, but most of these primary oocytes will degenerate by the time that a baby girl is born, and then the egg or the ovum at any stage from primary oocyte to the time of fertilization will be around 200,000. Out of those 200,000, we ovulate only 480 times between, let's say, 13 years old up to 60 years old, if so much. Now, the development of these oocytes then resumes in adolescence under the influence of several hormones. One is FSH, or follicle stimulating hormone. These will recruit monthly around 24 oocytes to complete meiosis 1. So before we develop these oocytes, then they are arrested in meiosis 1, and then they will start resuming this under the influence of FSH. And then these oocytes will start forming these haploid cells of an equal size. One that is going to be small, that is going to be the actual secondary oocyte, and then another one of large size that is going to be the first polar body. And this first polar body is going to disintegrate because it doesn't have the extra set of haploid chromosomes. The secondary oocyte, it will enter then meiosis 2. It will go as far as metaphase 2, and it will arrest until after ovulation. If this oocyte is not fertilized, it will never complete meiosis. It's only when the sperm cell fertilizes the secondary oocyte that it can complete meiosis. So you will have this alignment then of the chromosomes of the secondary oocyte in waiting for the complementary chromosomes coming from the sperm cell, so you can continue meiosis. So this is how then oogenesis and follicle development happens. So before birth, you have mitosis of this oogonia, and then you form these many oogonia, and then you arrest these cells in meiosis 1 into what we call primary oocytes. And in the ovaries, what you can see is these cells that look very similar in size and in structure. Once we born and under the influence of FSH, these primary oocytes can resume meiosis. So these will become, under the influence of FSH, these primary oocytes into secondary oocytes. And if you see on this part, the secondary oocytes are going to be much bigger. So they will have this large size, and then these cells here, granulosa cells that surrounds these primary follicles, or these primary oocytes, will become 2 layers. And then you also will secrete a membrane that is called the zona pellucida. And then these secondary oocytes will continue, and then they will become ovulated. Now, you also form a secondary structure that is called the first polar body. This will die because it doesn't have the chromosomes. And then this secondary oocyte, which is going to be arrested with its chromosomes in metaphase 2, of meiosis II, of course, if it's fertilized, will continue and resume meiosis II. But if not, it will die similarly as to a structure that we call the second polar body, which is not going to have the chromosomes. So first and second polar body are not going to have the chromosomes, but the secondary oocytes and the primary oocytes, they're going to have these chromosomes. And then these secondary oocytes will undergo changes under the influence of FSH and estrogens, in which the cells that surround the oocytes will secrete a fluid, which is called follicular fluid, and this will deposit this fluid into the antrum, or this space, and then you will have increased number of cells around this secondary oocyte into what we call the theca interna and theca externa, that is going to start producing the different hormones of the ovaries. And then on day 14 of the menstrual cycle, you will release these oocytes under the influence of LH. And if pregnancy happens, you will maintain for a while this structure that is called the corpus luteum, that is going to secrete progesterone. So this is primordial follicles and primary oocytes without any difference, and then you have these secondary oocytes inside these primary follicles, and the tertiary as well, more enlarged, with this antrum filled with fluid. So this is the oocyte surrounded by these granulosa cells, the corona radiata in here, zona pellucida in here, and then these theca cells, interna and externa, that are going to form these different types of hormones, like estrogens. So this is the diagram to show you what happens during the menstrual cycle or the sexual cycle. So regularly the number of days is supposed to be 28, with days 1 through 5 corresponding to the menses, and then days 1 through 14 previous to ovulation into what we call the secretory phase for the uterine cycle, and follicular phase for the actual ovary. And then after day 14, which is when ovulation happens, you have the days between 14 through 28, which will correspond to the luteal phase or the secretory phase of the uterine cycle. And these are the hormones that influence the development of the primordial follicles. So you have FSH, follicle stimulating hormone, that is released by the pituitary gland, anterior pituitary gland, that makes these primary follicles to grow into secondary and then into tertiary. And then with the surge of LH, this mature oocyte will be released at day 14, and then corpus luteum will form, secreting progesterone. If pregnancy happens, this will last for a few weeks, but if not, you will have involution. So this will shrink, and then you will have what we call corpus albicans. And then you start the cycle again with new primordial follicles. You start again FSH high, and then kind of getting low, a surge of FSH here at day 14 together with LH, and then maintain low levels during this luteal phase. So hormonal control of ovulation happens because as this follicle is growing, you have these thecal cells making estrogens or estradiol, which is going to activate anterior pituitary gland and hypothalamus. Hypothalamus is going to secrete GnRH, or gonadotrophin releasing hormone, and anterior pituitary gland is going to secrete LH and FSH under the influence of estrogens or estradiol and GnRH. And then this LH and FSH will help in the growth of and recruitment of more primordial follicles and in the release of this oocyte. And then the cycle will start over again. This is an endoscopic view of ovulation. So all of this happens in the cortex of the ovary. And then you have this follicular fluid released also into the pelvic cavity. For the menstrual cycle, it's the cycle that consists of the buildup of endometrium during most of the sexual cycle, followed by the breakdown and vaginal discharge. It's divided into 4 phases: proliferative phase, in which the endometrium is proliferating or growing; secretory phase, in which the endometrium is releasing glycogen; premenstrual phase, before the menstrual phase; and the menstrual phase, in which you have the discharge of this endometrium through the vagina. And then whenever the female has this blood coming from the vagina, this will be the day first of the sexual cycle or the menstrual cycle, which in average lasts hopefully 5 days. So changes that happens during proliferative phase. You have rebuilding of the functional endometrium that was lost in menstruation. So at day 5, your endometrium is about 0.5 millimeters of thickness, and it has only the basal layer. And then under the influence of estrogen, which is a growth factor, it's a growth hormone, you have mitosis of this basal layer, so you start having regrowth of the functional layer and also the blood vessels. And then by day 14, the endometrium is like 1/8 of an inch or 2 to 3 millimeters in thickness. And then the estrogen also will stimulate the endometrial cells to produce progesterone receptors, just in case that the female get pregnant. And so you can maintain a good thickness of the endometrium to nourish the fetus. So in secretory phase, the endometrium, so that will be after day 14, so between day 15 to 26, the endometrium will thicken more in response to progesterone that it is released by the corpus luteum. And then you not only have that, but you have thickening of this endometrial plumpness because you have secretion and fluid accumulation rather than mitosis. And then again, your endometrial glands will secrete glycogen, and then you have 2/8 of an inch or 1/4 of an inch of thickness of the endometrium, and this will be enough for harboring the fetus. Premenstrual phase is the period of endometrial degeneration. It will last 2 days of the cycle, and in this case your corpus luteum undergo atrophy, so you're having low levels of progesterone. So one of the things that is going to trigger for menses is the drop of progesterone, which was one of your key points. And then you will trigger, because of this, a spasmodic contraction of the spiral arteries, which is going to lead to ischemia, hypoxia within this endometrium, and then the menstrual cramps, as I told you, and then necrosis of these endometrial cells. And then when you are at day 28, you start the discharge of this endometrium, which starts the cycle into what we call the menses. Average bleeding during during the menses will be 40 millimeters of blood and 35 millimeters of serum fluid over a 5-day period. So this is the changes of hormones and the changes of the endometrium during the uterine cycle or the sexual cycle. So in these first 14 days, we have proliferation. of the endometrium. This is called proliferative phase. Estrogen levels goes up because it's a growth hormone that is going to allow this shed of these cells to regrow again. And then as this is happening, you have also enlargement of these endometrial glands and an enlargement of these spiral arteries. And then progesterone levels are low, but they peak a little bit around day 13, similarly as to estrogens. And then during the secretory phase, which is after day 14, the ovulation period, you will have a surge of progesterone because you have the uh, corpus luteum secreting progesterone, and you maintain kind of high levels of estrogens. And with that, you will have secretion of glycogen of these endometrial glands, and you maintain a thickness of the endometrium just in case the pregnancy happens or this ovulated egg get fertilized. But if that doesn't happen, then you will have your premenstrual phase in which you have hypoxia of this tissue within the endometrium, and then you start the menstrual cycle again. So these are the arteries, how they penetrate the endometrium. This is the endometrium with these glands in here, and then these spiral arteries will enlarge during proliferative phase together with these glands. And then during secretory phase, these glands that are straight, they become a little bit tortuous, and then they start secreting. And then if nothing happens, no fertilization happens, these arteries undergo constriction. They create hypoxia, necrosis of this tissue, and then the cells, they don't of the glands, they don't connect into the lumen, and then they regress in size. Okay, so the breast or mammary glands are part of the reproductive tract, or sorry, system in females. They are amount of tissue overlying the pectoralis major muscle that under the influence of estrogens during puberty, they enlarge in size. Now, the mammary gland develops within the breast during pregnancy as well. And then they will remain active in the lactating breast. And then they undergo atrophy when the woman ceases to nurse. The anatomical regions of the breast consist of a body, which is a conical area that ends in the apex, which is the nipple. Then on the lateral superior portion of the breast, you have the tail, axillary tail, that is the extension towards the armpit. And this is where you have lymphatics that are going to connect to the to the lymph nodes within the axillary area, the axillary lymph nodes, and this will be an important route for breast cancer cells in metastasis. The nipple is the area that it is surrounded by a dark skin that is called the areola, and in this area you have capillaries and nerves that are more sensitive. And then in here you also have these sensory nerve fibers that helps to trigger milk ejection when the female is nursing a baby. Within this tissue we have glands that are called the areolar glands that are intermediate between the sweat glands and the mammary glands. And actually the mammary glands are coming from the integumentary system. Now, we have within this tissue smooth muscle fibers in the dermis of the areola that will contract in response to cold touch and sexual arousal that will wrinkle the skin and erects the nipple. Now, in non-lactating breast, most of the mammary gland is made out of adipose tissue and collagen fibers. And then the breast size is not determined by the actual amount of lobules in the female. So in the female breast. So we have all females, we have between 18 to 20 lobules or segments of the breast. But the amount of adipose tissue is different. So if a female has a large breast size, it has more adipose tissue than others, but the lobules is the same. And then the breast is held into the anterior wall of the thoracic part of our body by the suspensory ligaments, which are nothing else but collagen fibers that connects to the pectoralis major. The glands inside the lobules of the breast connects into the nipple through ducts. And we have, as I told you, between 18 to 20 lobes. Your book talks about 15 to 20. And then these lobes are surrounding the nipple area. And then each lobe will drain milk into the lactiferous duct. And then this lactiferous duct will have a dilated area next to the nipple that's called the lactiferous sinus. So many lactiferous sinus then, or sinuses, receives this milk from the glands of the lobes, and then the milk will be ejected. Now these ducts ends in the acini or sacs within the glands, and these will be surrounded by these contractile cells that are myoepithelial cells that respond to oxytocin. So this is the longitudinal section of a breast in lactating state. So these are the lobes separated by these suspensory ligaments. And then here we have the acini. This will have these cells that produces the milk, and this acini will be surrounded by myoepithelial cells that will contract under the influence of oxytocin. So when the baby starts stimulating the nipple, sucking the nipple, the female is going to have this feedback mechanism where she's going to secrete oxytocin from the posterior pituitary gland, and this oxytocin will reach these myoepithelial cells, so it will contract the acini, and the acini will drain the milk into these lactiferous ducts, which are going to merge into the area posterior to the areola in the nipple, and then you have these lactiferous sinuses that will collectively receive the milk and drain into the ducts of the nipple. And then this is the areola with the areolar glands. This is a sagittal section to show you again this acini and these lobes, and then the suspensory ligaments, how they connect into these pectoralis major, and then you have these fat cells, this adipose tissue that makes the bulk of the breast. And this is a magnification of this acini and these myoepithelial cells. So this is then the end of this chapter. If you have any questions, you can email me. And have a good day or have a good night. Bye-bye.